Hello everyone, welcome back to Dr. Ghosh's Meet channel. The current topic that we are going to go for, it is a very unique popular talk. And the popular talk that is based on the current advances on recycling approaches of xenobiotics compound from soil and water bodies. The entire project has been conceptualized by Dr. Dipankar Ghosh, Associate Professor and PI of Microbial Engineering and Algal Biotechnology Laboratory, Department of Biosciences, JS University, Kolkata. First of all, I would like to acknowledge to all my MICROBIALs, especially my PhD and master students. And I would also like to thank three students, Shayon, Pomita and Rituparna, for their immense effort to summarize this assignment to get completed. So let's get started the journey of this popular talk. So we will be talking about xenobiotics and their impact. So xenobiotic is a comprehensive term which is actually being derived from Greek word xenos, which so-called means the foreigner or the stranger, and bios or bios means the life, which is used to indicate a man-made foreign chemical substance which is found within an organism that is not naturally produced by or expected to be present within that organism. Indeed, it is not recognizable by the enzymatic system of living organisms, and if introduced into the environment, it can accumulate and reach certain concentration to cause undesirable effect. So we can take some examples considering the organic xenobiotics, which include pesticide, fuel, solvents, alkane, polyaromatic compound, chlorinated and nitroaromatic compound, azo compound, dyes, organic sulfonic acid, nitrogen and phosphorus components. In contrary, you can also say there are certain inorganic xenobiotics do exist, like toxic heavy metals like cadmium, copper, nickel, chromium, zinc, arsenic, some synthetic fibers, polymers such as polyethylene, polypropylene, as well as plasticizers and softeners, which usually being used in different textile industries. Xenobiotic pollution of environment is a global concern, which usually caused by anthropogenic activities such as urbanization, overpopulation, expansion. The prominent substances such as polycyclic aromatic hydrocarbon, heavy metal ions, pesticide, fertilizer, oil derivatives are found in soil and water. Dioxins, dichlorophenyl, trichloroethane, polychlorinated biphenyls, polycyclic aromatic carbons, nitroaromatic compounds are preliminary threat to the soil ecosystem and dynamics of developed nations. Benzene, nitrobenzene, ethyl benzene, toluene, xylene, aniline, even chlorinated solvent also acts as a xenobiotic compound. Cosmetic and personal care product also contribute as a xenobiotic pollutants, especially parabens in soil and air and azo dyes in soil. From agriculture field perspective, where multiple rounds of pesticides and different xenobiotic fertilizers are used in larger amount, which actually destroys the water quality and aquatic plants and animal life cycles. So now we are going to talk about impacts of xenobiotics on soil ecosystem. Physicochemical characters of soil are altered by xenobiotic compounds. It is a very well-known phenomenon. They influence the soil pH, moisture, water holding capacity, organic nitrogen. The overall texture of soil can be having a high yield impact caused by the introduction of xenobiotics. These all factors of soil affects the microbial activities in soil and their dynamics that promote the soil quality and also reduce the plant growth. The xenobiotics create ecotoxicological effect in soil, which causes by natural and the synthetic pollutants that affects the animal along with human, vegetation and microbial regime in an implicative way. Organic xenobiotic like fertilizer, phosphorus compound, potassium, nitrogen, even zinc metals. There are certain pesticides. They also produce unexpected growth of plant in water, or it can also induce the phenomena of eutrophication. Due to extensive eutrophication, the amount of dissolved oxygen becomes decreased. Demand of biological oxygen demand will elevate it. So by increasing reactive oxygen species alter the homeostasis in fishes, cause oxidative stress, which creates a mess in aquatic food chain and ecological balance. So these are the few pictorial presentation which shows that how xenobiotic compound is influencing the normal life habitats within the water bodies and altering the water ecosystem. Now we need to talk about the recycling approaches of these xenobiotic compounds. So as we have seen already, xenobiotic compounds are creating huge ill impacts on the soil as well as in the water body and their associated ecosystem. So that needs to be eradicated, or that needs to be somehow get resolved. So now we are going to talk particularly to find out these solutions. Those are called the recycling approaches of these xenobiotics. So first is the physical degradation. The way of degradation of xenobiotic compound can be categorized mainly in three parts. As I told, first one is the physical degradation. Physical treatment of xenobiotics can be defined as the thermal or non-thermal treatment. So some processes for soil includes the electric resistance heating, ERH, steam injection, vitrification techniques, incineration, pyrolysis, even water flushing also used to reduce pollutants, especially xenobiotic pollutants from the soil. So these are mostly confining the physical degradation approaches. For the water treatment, physical degradation includes screening, communicator, sedimentation approaches, and heat treatment. So as you know that each and every technique do contain or do bear some merits and demerits. So physical degradation also having different advantages for recycling the xenobiotic compounds. Like the contaminant toxicity is checked by this technique. It is used to remediate the soil contaminated with the explosive and hazardous waste. It can create treatment of organic contaminants in soil and oily sludges. Whereas it also contains certain disadvantages, like metals are not destroyed by this approach, and it ends up with the flue gases in the ashes. Rocky soil need to be screened before use, and drying of the soil required for the wet soil. Most of these cases, solid compound cannot be degraded properly. They can only segregate it from the soil and water. So the next recycling approach for xenobiotics is a chemical degradation. In soil and water, several heavy metals are present, such as cadmium, lead, mercury, iron, copper, arsenic. So these heavy metals can be separated by electrokinetic process, in which cathode and anode electrodes are being used to separate positively and negatively charged xenobiotic constituents. So this is a nice pictorial presentation which have been showing how electrokinetic remediation of soil can be performed by using the fuel cell technology, by using anode and cathode. And the third most interesting recycling approach of xenobiotic constituents is the bioremediation. Bioremediation refers to the process of using organic or living organisms, including plant roots, and diverse ranges of microorganisms that will reduce or remove the environmental pollutants like toxic wastes found in soil, water. This process can be separate or segregated by two different sub-avenues, likely microbial remediation and phytoremediation. Microbial remediation mostly focusing on the involvement of microorganisms, their physiology, and metabolic reactions. Microbes has an incredible capability to catabolize with the help of different genes, enzymes, to execute the degradative pathway that will reduce or break down the xenobiotic constituents. Multiple rounds of microorganisms, including Pseudomonas, Bacillus, Alkaligenes, Cellulomonas, Microbacterium, Micrococcus, certain purple non-sulfur bacteria like Rhodococcus, some fungi like Aspergillus, Penicillium, Trichoderma, some fruiting bodies like Streptomycin, Rhodotorula, some mold like Candida. They can also perform by using their metabolic activity to reduce the load of this xenobiotic compound. Most predominant microbes are Pseudomonas. They can degrade, break down several organic compounds like hydrocarbon, polyphenols, organophosphates, polychlorinated biphenyls, and polycyclic aromatic naphthalene compounds. Pseudomonas species contains xyl for xylene, NH for naphthalene, sal for salicylate, CAM, plasmid that will allow to express certain rounds of enzyme that will elevate the degradation of camphor xenobiotic components. Bacillus subtili strain can able to degrade petroleum hydrocarbon. Even strains of Bacillus thuringiensis, Bacillus subtilis involves in the breakdown of xenobiotics from the rhizospheric zone of Panicum aquaticum. Polylactic acid degraded by Bacillus pumilus B12 strain, that is the second most common biodegradable polymer found in commercial plastics. In the bioremediation section, another sub-avenue which were discussed like phytoremediation. Phytoremediation is an economically and environmentally sustainable technique as it utilizes green plant to contain, sequester or detoxify contaminants from contaminated soil or water bodies. Therefore, it is one of the most significant parameters affecting the behavior and the linked toxicity in terrestrial and aquatic ecosystem. Phytoremediation utilizes many mechanisms that will accelerate or speed up the degradation process for removal or immobilize the pollutants, including degradation like rhizodegradation or phytodegradation, accumulation, that is phytoextraction or rhizofiltration, dissipation like phytovolatilization, immobilization like hydraulic control or the phytostabilization. Usually, the contaminated groundwater can be bioremediated through the phytodegradation, phytovolatilization, rhizofiltration, rhizodegradation, and phytodegradation process. Whereas the surface or the wastewater contamination can be treated by rhizofiltration, phytodegradation, and rhizodegradation approaches. Soil sediments or the sludges contamination are remediated through phytoextraction, phytodegradation, phytostabilization, rhizodegradation, and phytovolatilization. In many cases, plant species, diverse ranges of plant species cannot perform well. They need to aid or enhance the phytoremediation efficacy. Such aid include the biochar, EDTA, endophytic bacteria as a helper microbial entity, along with the association with the plant root. So let's talk about certain definition that is actually involving in phytoremediation mechanisms. The first one is the phytoextraction. Phytoextraction is a process in which the contaminants are taken up by plant root, resulting in their accumulation in above ground parts. Therefore, can be harvested and ultimately disposal of plant biomass can reduce the environmental pollution in the soil. Phytostabilization. This remediation can be achieved through the inactivation or immobilization of toxicants or pollutants within the root or in the rhizospheric zone. Third one, rhizofiltration. Rhizofiltration involves removing toxic substances using plant root in a saturated zone to remediate aqueous contamination in the wastewater, in the groundwater, or in the surface water through adsorption and precipitation onto roots. So what is phytovolatilization? In this process, water-soluble contaminants are taken up by means of plant, resulting in discharge of volatile contaminants or release of non-volatile degradation products. Phytodegradation is another round of phytoremediation mechanism in which metabolization or metabolic degradation of the organic compound is done in soil surface, even in the groundwater, under sediments or sludge through enzyme produced by the plant. Rhizodegradation includes the augmentation of degradation that is taking place naturally in soil by influencing the plant root that can lead to the ideal detoxification of these xenobiotic contaminants. So this is a nice tabular presentation which shows the different columns having mechanism, common plant name, what are the different xenobiotic contaminants can be eradicated, having with their advantage and disadvantages. You can see the phytoextraction, phytostabilization, rhizofiltration, phytovolatilization, phytodegradation, rhizodegradation, all involving different kinds of plants. It could be lettuce, tobacco, corn, Indian mustard, water starwort, water moss, mustard, broccoli, rabbit food grass, and they are allowed to eradicate or recycle different forms of xenobiotic compounds, including different heavy metals like nickel, cobalt, lead, zinc, copper, mercury, arsenic, uranium, selenium, including DDT, petroleum, textile azodyes, petroleum, and also dichlorophenols, including different advantages and disadvantages. So usually advantages are including least environmental disturbances. It used on the large range of contaminants, cost effective for large contaminated sites. Whereas the contaminants may enter the food chain through the animal which eat the plant which already absorb those toxicants. It is also limited to soil less than 1 meter from the surface and groundwater less than 3 meters from the surface. So these are the few constraints which are involved in these processes that needs to be rectified or that needs to be altered in a way that it could be showing more influence to advancement or to reduce or to increase the potentiality of this approach. So we'll come to a conclusion. So xenobiotics have been one of the major pollutants in both soil and water. Microbial degradation of xenobiotic assumes significant since it provides an effective and economic means of disposing of toxic chemicals, particularly the environmental pollutants. Different forms of organic molecules like pesticide, fuel, nitrogen, phosphorus compounds, even some including some inorganic molecules like toxic heavy metals, synthetic fibers, affect both soil and water can be eradicated. Different approaches that are eco-friendly and non-costly, industrially feasible techniques that have been employed to degrade these xenobiotic compounds to reduce their load in soil and water. The employment of these techniques provide a good soil water quality without affecting the ecological dynamics in the microenvironment. The enhanced crop yield can also be achieved by improving the soil quality through these processes and their implementation that will allow concomitant degradation of xenobiotic. The increasing discovery of new xenobiotic degrading plant system, enzyme system, metabolic enzymes, more potent microorganisms that can open up a new door to prevent the xenobiotic pollution in both terrestrial and aquatic ecosystems. So these are the few references for further studies. If you are really interested, you can come across these references to gain more knowledge, more ideas about the genetic aspects or enzymatic or metabolic aspects of these remediation techniques which are involving in plant as well as in different microbial systems. So I believe you enjoyed this session. You get a lot of informations in a nutshell, and that will increase your interest. You can gain more knowledge by following further reading section. And I definitely believe it enlightened you to get more research to be done, and that will allow you to nurture more knowledge for your future endeavor. I hope you enjoyed. Thank you for listening and being patient with my deliberation. Thank you very much. Good luck. Thank you.